Hey everyone, and welcome to today's Explainer. I am seriously so excited to share this one with you because we are tackling what has to be one of the absolute coolest biological whodunits in all of human history. I mean, have you ever looked at a monkey effortlessly swinging from a tree and thought, why don't we have one of those built-in balancing ropes? Well, today we are piecing together an amazing scientific detective story, the mystery of exactly how humans lost their tails. We're going to dig into microscopic embryonic evidence, deep fossil history, and even the genetic dark matter hiding right inside our DNA to crack this case wide open. So get comfortable because we've got some fantastic visuals to walk us through the whole thing. All right, let's dive right into it. The core question driving this entire investigation is pretty simple on the surface. Why are humans the only primates out there without tails? It sounds like a basic question, right? But the answer has actually eluded scientists for generations. It's a trait that fundamentally separates the hominoids, so that's us and the great apes, from the rest of the primate family tree. And to solve this, we can't just look at modern humans. Nope. We have to follow the evolutionary breadcrumbs back millions and millions of years. So our journey starts way, way back in the deep past. Picture this, 33 million years ago, our ancient primate ancestor, a guy named Egyptopithecus zeuxis, sported a really prominent long tail. But roll the evolutionary clock forward to about 25 million years ago, and something absolutely monumental happens. The hominoid lineage splits off from ancient Old World monkeys, and right at this massive evolutionary crossroads, the external tail just vanishes. Fast forward to today, and all that's left of that once majestic appendage are 3 to 4 tiny fused vertebrae sitting right at the base of our spine. You know it as the coccyx, or your tailbone. Now, a lot of researchers think this profound structural shift was absolutely crucial for bipedal locomotion. Basically, losing the tail is what helped us stand upright and walk on two legs. But how exactly did we lose it? Well, that brings us to our first major clue. Check out this quote from researchers who are analyzing the Kyoto collection of human embryos. It reveals a phenomenal plot twist, and honestly, I absolutely love sharing this fact. It reads, although the human tail is completely absent at birth, the embryonic tail is formed just as in other tailed amniotes. And by amniotes, they mean reptiles, birds, and other mammals. Just stop and think about that for a second. You, me, literally every person walking the earth right now, we all actually grew a tail while we were in the womb. We didn't just evolve away from growing a tail entirely. Our bodies still start to grow one, and then they actively eliminate it before we're born. To figure out how that wild disappearing act works, scientists had to look at our embryonic development closer than they literally ever had before. To track this disappearing embryonic tail, scientists keep a close eye on something called a somite. Basically, these are little blocks of embryonic tissue that eventually grow up to become your vertebrae, ribs, and skeletal muscles. Think of them as the tiny architectural building blocks of the spine. So if you want to know what on earth is happening to the tailbone during fetal development, you have to count the somites. For decades, it was ridiculously hard to see and count every single one of these in a tiny developing embryo. But today, thanks to some incredible modern 3D reconstruction tech, researchers can map them out perfectly. Now, what's really fascinating about this data from a 2018 study by Tojima and colleagues is the exact timeline it lays out. It documents the number of somites across different phases of human embryonic development, which are known as Carnegie stages. By meticulously counting the percaudal somites, so those are the ones in the main body, and the caudal somites, the ones specifically in the tail, these researchers managed to track the precise moment in development when the tail vanishes. It is literally like watching a time-lapse of our own evolution playing out over just a few weeks of gestation. When we look at the progression of those caudal somites, the tail builders, the pattern is just undeniable. Right at Carnegie stage 15, we have about 12 pairs of them. By stage 16, this peaks at up to 16 pairs. I mean, the tail is actively growing. But then, look what happens at stage 17. Boom, a sudden massive drop down to around 9 somites. In an absolute flash of developmental time, roughly 5 pairs of these spinal building blocks are just completely wiped out. So the obvious question is, what exactly is destroying them? Let's break down how this physical disappearance actually goes down. First, the somites grow and hit their peak at stage 16, just like we saw. But then step 2 is where the magic, or I guess the demolition, happens. The embryo triggers a biological process called apoptosis. Now, apoptosis is programmed cell death. Essentially, a microscopic cleanup crew of cells called macrophages swoops right into the tail region and starts actively dismantling the tissue. Then, by step 3, the caudal somites rapidly decrease as the cellular demolition team completely clears out the embryonic tail by stage 17. So we don't just forget to grow a tail, our body executes a highly precise, targeted demolition of it. All right, so we know when the tail disappears in the womb, and we know how it disappears physically, thanks to apoptosis. But why does the human body suddenly call in this demolition crew? What's giving the order? To answer that, we've got to zoom in even further, way past the cells, right down into our DNA. We are looking for the genetic smoking gun here. And recently, researchers with the Dark Matter Project at NYU Langone Health finally tracked down the culprit hiding in a gene known as TBXT. But the culprit wasn't a missing gene. No, it was actually a tiny genetic hitchhiker called an ALU element. This is a short, transposable stretch of DNA. You can kind of think of them as genetic jumping genes that just copy and paste themselves all throughout our genome. Sometime around 25 million years ago, one of these little ALU elements jumped right into the DNA of our shared hominoid ancestor. And, well, it changed primate history forever. Here is exactly how it all went down. This specific ALU element inserted itself into a non-coding region of the TBXT gene, which is the gene that controls tail development. But crucially, it landed right next to an older ALU element that was facing the opposite direction. These reversed elements actually bound together, creating a literal loop in the RNA. And this loop caused the cell to just skip over a key part of the gene, an event scientists call hominoid-specific alternative splicing. This tiny, microscopic loop is the exact mechanism that stunted our tails. I mean, what an absolutely elegant and mind-blowing discovery. Now, understanding this whole genetic and embryonic sequence doesn't just solve a massive evolutionary mystery, it also solves a medical one. We need to pivot just a bit here to talk about a very rare but very real congenital anomaly. Babies born with what looks like a human tail. For over a century, the medical community held a very specific belief about what these appendages actually were. But our new 3D embryonic mapping, it has completely debunked it. And this brilliantly illustrates how advancing technology corrects historical science. Back in 1901, a researcher named Harrison came up with a hypothesis. He basically said that a baby born with a soft, boneless pseudotail was simply holding onto a vestige of a boneless tip of the embryonic tail, something he called the caudal filament. For well over 100 years, medical textbooks just repeated this. But in 2018, Dr. Tojima's 3D reconstructions definitively proved Harrison wrong. The new imaging clearly showed that caudal somites, those bony building blocks we talked about earlier, actually extend all the way to the absolute tip of the embryo's tail. There is no boneless caudal filament, ever. Therefore, the soft pseudotails occasionally seen at birth cannot possibly be a retained vestige of this embryonic phase. It completely shifts how the entire medical field views this rare condition. Okay, so we have the timeline, we have the embryonic physical process, and we have the exact genetic code that executed the change. But, as we know, nature rarely gives us something for nothing. Evolution is essentially a giant game of compromise. So, as we wrap up our investigation today, we have to look at the motive and, maybe more importantly, the cost. What ancient price did we actually pay for our tailless, upright bodies? So the crucial point to take away from all this is our bipedal locomotion, our amazing ability to walk completely upright, which freed our hands to use tools, build fires, and create entire civilizations, is deeply, fundamentally linked to the loss of our tails. We gained the world, for sure, but that TBXT gene mutation came with a severe adaptive cost. This ancient trade-off is actually linked to an increased risk of neural tube defects like spina bifida. The very genetic mechanism that sculpted our modern bodies also leaves our spinal cords uniquely vulnerable during development. It's an evolutionary choice made millions of years ago that continues to directly impact human health to this very day. It really makes you look at your own body a bit differently, doesn't it? Just think about it. A single jumping gene millions of years ago fundamentally altered the entire trajectory of our species. It cost us our tails, granted us our upright posture, but left us with a vulnerability we are still managing today. It really forces us to ask, what other ancient genetic trade-offs are hiding out there within our modern human bodies? What other jumping genes have quietly engineered the traits that we just take for granted every single day? I really hope this explainer gave you a deep appreciation for the incredibly complex detective work that goes into understanding our own evolution. Thanks so much for exploring with me today, and keep asking those big questions.